aseptic techniques, possible contamination, management, and biosecurity guidelines. This is the topic, and they all are totally connected. So the main objective of this talk is, first of all, understand your enemies. Whose are your enemies in the laboratory? Number second, how the norms and guidelines should be proceed or followed according the standardization or the specific function of the lab, right? And the biosecurity labels, that is another very important issue, that every lab has to be designated as level 1, level 2, level 3, and level 4 laboratory. We call it biosafety level 1, 2, 3, 4. Okay, so that is also another thing. And the fourth one is how to work in a tissue culture lab, means best practices. Okay, the best practices of working in a laboratory. Okay, there are so many examples I can give you that we have faced a very critical contamination in a lab where you do not know what is the source of contamination, and you lost basically all your cultures. Imagine I got one plant material from China or India or Japan. That is the only plant material I had. It's got contaminated. You cannot imagine the impact you are going to have as a researcher or as a commercial lab, right? Because the material, conservation of the material, preservation of the material is extremely important. You protect them better than you protect yourself, because they become your life, they become your business, they become your project, right? So that these are the very, very important concerns. After the another things, that there are contamination that comes within that tissue, so you are not sterilizing it properly, right? So they are inside the tissue, and that contamination you will face within few days, because the bacteria, fungi will grow quickly in the nutritive media. But there are 90% contaminations that are injected by the bad practices. Means you are not following the best practices in the lab. You are not using lab coat, you are talking, you are sneezing, you are running close to the laminar airflow, you are putting or injecting and touching different kinds of objective. You are not using gloves, or gloves are contaminated. You are using pipetting inside the hood, and you are touching your headphones, listening the music, remembering about your novia and novia, okay, and working and cutting the flax plant, right? So these are not good practices, okay? So these kind of thing happens. UV light, you haven't switched on the UV light, or you have switched on the UV light and working in the UV light. That has happened with some of my friends, because they switch on the UV light and they start working in the UV light for 2 hours, and suddenly someone came and tell, hey, you are working in the UV light, and he was, oh my God, now I will become a mutant, right? So these kind of accidents are also common. Alcohol, for example, you know, ethyl alcohol, 70% alcohol that we use in lab, when you burn it, that is called cold burn. Okay, cold burn. Quemadura fría. Why cold burn? You can put on your skin the ethyl alcohol and put a lighter. It will burn you. You won't feel. Okay, so that is why it's called cold burn. So if you are working in a lab, working with the lamp, working with the fire, and just mishandling thing, you may lose your hair or skin, and you will not even feel until it completely degrade your epidermis. Then only you will feel it. That is why it's called cold burn. Working with the liquid nitrogen, it's another issue. Liquid nitrogen is very dangerous. Okay, very, very dangerous. The temperature is minus 192. So if you are not handling it properly, you can lose your eyes. Okay. I have been working with HCl, hydrochloric acid, 98% industrial grade. And once I drink it mistakenly. So my lips, my inner layer of the mouth, it was totally burned. Okay, I'm alive, obviously. And I had so many accidents, but anyways, you learn after the experience and you don't have to repeat it. Okay. Okay, so these are the global context of the topic. Okay, so if you think about whose are your enemies in the lab, there are so many enemies. Okay, but to understand your enemies, the sizes become very relevant. For example, everything in us, everything, any living organism and even non-living organism, everything is comprises or consists of atoms, right? Atoms creates the molecules, fluid molecules, for example, and these molecules become lipids and proteins. So the size of a virus, you can imagine, it's very similar like a protein. A protein could be of size of 6 or 7 nanometer, and a virus, it could be 10 nanometer. What is the size of COVID-19 virus? Anybody knows that? Okay, investigate. So for example, viruses are very small. They don't even can be seen by a normal microscope. You need to see them by electron microscope or a scanning microscope, right? Then we have flu viruses, which is little bit bigger, not little, like 10 times bigger than the polio virus. Then we have a smallpox virus, even bigger than that. And then we have bacteria, which could be up to from 1 micrometer up to 10 micrometer. Okay. And we have mitochondria, which could be even much more bigger than the viruses and even the bacteria in some cases. And after 10 micrometer, we have these cells. Okay. And they start from like 20 micrometer up to 100 micrometer, plant cell and animal cell. Pollen, obviously, it's going to be something that is bigger than 100 micrometer, right? And then we have frog egg. Okay. Very small, right? So you can think that these are the different size. And from here to here, you can see by only electron microscope, right? And from 1 micrometer up to 1 millimeter can be seen by the normal microscope, right? So how and why it is important to protect yourself with these bacteria? Because anything of it, anything, even the foreign particles, even any bacteria, fungi, viruses, you don't want them in the media, because media is rich. It's rich in nutrient. It rich is condition. It has light. It has water. It has everything that these bacteria or microorganisms require. So basically, they will grow happily. So if they grow in the media, what will happen? They will eat your plant and they will kill the plant. Okay. So basically, these microorganisms are not welcome at all in the tissue culture lab. Okay. There are wide variety of pathogens. Okay. So whether it is a pathogen or it is not a pathogen, you don't want both of them. Okay. Neither pathogen nor microorganism. Okay. You don't want them in a tissue culture lab, because if you are doing something in vitro, definitely it has to be free from viruses, right? That is why we use meristematic tissue, which does not have vascular system to produce plant, which can guarantee the absence of the viruses, right? And there are certain very prominent diseases that are very well known. That tomato is very seriously affected by Alternaria solani. It's badly affected by the leaf mold. So you never want these kind of pathogens, which are very specific for your crop that you are growing in vitro, right? So these are the pathogens that has to be avoided 100%. But even a simple E. coli, even a simple other kind of pathogen that just there to eat the media, okay, that is also not required, because eating the media, they will spread all over the plant, and at the end, they will eat your plant. If they don't eat your plant, basically they will cover your plant and you cannot do anything, because whatever you will do, it will replicate more bacteria or microorganism than your own plant. Okay. So that is the important fact. Sterile condition or aseptic condition means that something where you don't have any kind of microorganism, means free from life. Okay, so that is called aseptic condition. Okay. Contamination, as I already mentioned, it has two sources, main, internal and external, right? Internal can be cleaned, can be treated, okay, can be sterilized, okay, but it also depends on your good practices. But external, if you have a very clean explant, very clean conditions in the media, everything is sterilized correctly, but your external handling, how you are handling the media, you will contaminate it. I had a meeting with one of the very big laboratory in Germany, Vigro, and we had a meeting. So I asked them, how frequent is you have contamination? So they just turned back and start smiling with their technicians. And they told me, contamination? What kind of contamination? We never seen contamination. Okay. So I was really surprised by their reply, because they are much sure that things have been standardized so much. It's a very big company, and they are very master in what they do. So they think, they told me that if I have a contamination, just imagine that we are going to lose the millions of dollars in a single day. So contamination is not acceptable anyhow. Everything is totally guided, monitored, properly tagged, and connected and synchronized by computers. So whatever is entering in the lab and whatever is coming out the lab is 100% trackable. So that is why they follow these kind of guidelines, right? So they maintain it. And that is the idea. In the tissue culture lab, you should not have contamination unless you want them to grow within your plant, right? Just one example. If you sneeze in front of the hood means laminar air flow, just from the droplets, you are producing up to 200,000 small particles, okay, from your mouth, okay? When you kiss someone, within the case, we exchange more than 600 different kind of microorganisms, okay? There is a book that I published 3 years back, and we put that data in that book. Kissing someone is transferring, interexchanging 600 different bacteria, okay? So I'm not going to tell you that don't kiss, okay? This is not the idea, okay? I'm just giving you the idea that how many things you are going to exchange just from your saliva, okay? So the breathing, the sneezing, the talking, anything is full of bacteria, okay? So you don't want it. Contamination. What is the most frequent contamination? Contamination, the most frequent is bacteria, E. coli, okay? Staphylococcus. These are the bacteria which are everywhere. They are in the air, they are on your clothes, they are on your skin. So basically, whatever you move inadequately, they fall down in your media and will become grow. How long or how many days they will take to grow? Media should be tried. You can put a media on an incubation chamber before putting the plant, just to check out whether your media is properly or the flask or anywhere you have mistakenly entered the bacteria or microorganism inside your flask. So you can put the media in an incubator, okay? Some lab, they do this, we do this. So basically you put on 30 degrees centigrade for 2 days, come out and take a flask. Oh, there's no bacteria, fantastic. Because the best temperature for bacteria to grow is more than 30 degrees when it is warm. So if 30 degrees centigrade for 2-3 days, you have no contamination, congratulations, you have prepared media very well, right? And if you have the bacteria inside there, in 24 hours, you will start seeing the microorganism there, right? So the bacteria is the most important factor everywhere you have it. And this is depending on the bacteria, it could be black, it could be red, it could be blue, it could be any color of, but it is going to be, you see the layer over here, the whitish layer. This is the E. coli, for example. Okay, so they start growing here. Now you can see the tissue has necrosis, so it become black. They become less yellow, less green, and after few days they will totally destroyed. Okay, so bacteria, this is how it grows. If it is fungi, worst, because fungi is very dangerous because it creates spores, and when it creates spores and you mistakenly open the flask, inside the incubation room, what will happen? The spores will spread everywhere in the air, and you will don't even see them. And they will get, whenever you open it, whenever you have the airflow, they will move all around the lab, and they will keep contaminating for a long period of time. So you will never stop the contamination. Okay, are you getting my point? How delicate and sensible is working in a tissue culture lab? Okay. So bacteria, for example, they have a very specific characteristics. We call them ooze. Okay, ooze is like a point. Okay, ooze like is a point, and sometimes when they grow much, you can find some layer. Okay, they could be of many different kinds of colors. Sometimes they even look beautiful because pink and yellow, and they have beautiful colors, but they are something you don't want in the lab, right? Fungi is the second one. But fungi, when it comes, it's very difficult to eradicate them, right? Normally, they have a fuzzy appearance, right? Fuzzy appearance, you understand the fibrosa, right? All of them, they produce by these spores, okay? Most of the time, fungi created in the humid areas. If your lab is not good dry conditions, it will be anywhere on the floor, on the walls, on the cleaning room, or from outside, from the soil, okay, or even within the tissue. Okay, a fungi looks like this. This is a normal fungi mildew, and you can see that how it grow. Okay, and it's grow within 1 day, 2 day, it will cover your flask. Okay, they grow extremely faster. The third one is virus. Now, how would you understand, or how would you identify whether your plant, your tissue culture condition has a virus particle or not? There is no way to know it. There is no way, because virus do not create colonies like bacteria or fungi. Okay, and you don't have electron microscope, so you can see it. So normally virus is already inside the tissue. Okay, so that is why if you want to create a virus-free plant, you have to use the meristematic tissue. So on the apical meristematic tissue, or the bud, you have to cut the smallest part of that bud that has the major, in a major part that must have meristematic tissue, means no vascular system. So since it was not connected with the vascular system, certainly the virus, even the plant is infected, it is not in the tissue. So in some cases, what do you do? In some cases, people even take the tissue to the electron microscope laboratory to find out whether they observe any kind of virus particle. Or in some cases, they, from starting the material from it, they take out the DNA and check for the genomic sequencing if they have any kind of sign of any kind of pathogenic viruses in there, right? So that is another way, okay? And the third way is basically just do the virus indexing when you have your final product. So before selling the product, you have to go to the virus indexing. You have to get a certification by molecular biology lab, where they do the southern hybridization, PCR, RT-PCR, the Western blot. So there are DNA sequencing. So there are many different techniques established for different kind of viruses. So if you want to sell it, then you have to have the certificate for that plant. Okay, lot. So viruses are almost impossible to identify. So that is why you have to use molecular techniques. Okay. Some cases there are heat treatments. You can put your X plant in a certain temperature for a certain period of time in a growing condition. And we know that after a few days, the virus, if it is there, it will be killed. Okay. So these are the another methods. In latent contamination, latent contamination is something very, very difficult, because that is very related with the viruses, but sometimes in bacteria also, because you have the bacteria inside, and within 1 week, you see everything growing great, no sign of contamination. But maybe after 3 weeks, suddenly you have the contamination. So you will be surprised that what happened. The flask was completely closed. You don't have even opened it. Why? There is contamination. Okay, so this kind of contamination called the latent contamination that not suddenly reflects, suddenly appears. They take a longer period of time. So that is why in most of the lab, it is recommended that you test your media in an incubation chamber for at least 1 week, okay, to avoid any kind of uncertainty that you can have a latent contamination, okay. The labs generally are extremely clean, okay. They are cleaned, well balanced, and the mobility of the people is very controlled, okay. So all the people entering and coming out, they are totally covered, okay. I will show you some videos where you can understand how the process going on depending on the level of safety, okay. 1 2 3 4, okay. So anyways, this is the basically the general introduction and some example, but the whole process of maintenance of the aseptic conditions, it does not come just because of the best practices, but also by some techniques, okay. And there is a technique that is called sterilization. Sterilization is a technique by which you can kill microorganisms in your vessels, in your flask, in your media, okay, whatever you are going to use in chemicals, okay, on materials. So sterilization can be done by different techniques, different methods. It could be by proper combination of heat, okay. It could be chemicals. It could be irradiation by rays. It could be high pressure. It could be a simple filtration. So there are different ways depending on what you want to sterilize, right? For example, filtration can be used for the heat sensitive material like hormones. You cannot autoclave hormones. You will destroy the hormone. You cannot sterilize vitamins, right? By autoclave, you will kill the vitamins, right? So basically, the filtration technique is used for the chemicals that are heat sensitive, okay? Rest of it, whether it is a glass vessel, whether it is the steel material, without any problem, they could be autoclave, and they can be autoclave by the irradiation also, right? Heat autoclave or chemicals, they are used to sterilize your explant material, okay, because explant you cannot autoclave, explant you cannot filter. So explant plant material that you are using in tissue culture, it has to be treated by different kinds of chemicals, soft chemicals, okay. If you use something very toxic, obviously you are going to kill the plant, right? So sterilization is making something complete free from organism, right? And we call a culture that is called exonic culture. So if someone asking, okay, tissue culture is all about exonic culture. Exonic culture, germ-free, uncontaminated, free from pathogenic and non-pathogenic microorganism, contain only one organism. And what is that?
The plant?
Yes. Only your plant, only one organism that is being cultivated. A culture is free from any kind of organism. It is not contaminated by any, and we call it pure culture, okay. So exonic culture is your meta, it's your objetivo, okay, in tissue culture. So sterilization basically not because sometimes bacteria, fungi, not sometimes, most of the time, their growth is not only an issue. They also produce secondary metabolites, okay. Bacteria and fungi, they create number of compounds. Penicillin, it comes from a fungi, right? It's an antibiotic. So there are so many biomolecules that are produced when bacteria are happy, reproducing, producing. So what will happen? They will affect your plant by the secondary metabolites, okay. So the source of the contamination, they are everywhere. It could be your culture or explant. It could be your vessel. It could be your media. It could be your instrument you are using. And it could be anything that is coming from the environment, okay. So there are many sources of contaminations, okay. In aseptic techniques, we have two kinds of treatment. I already mentioned the chemical treatments and the physical treatments, right? Chemical treatment, they are disinfectants, antibiotics, okay, and sublimate, other kinds of chemicals. Now, disinfectants, it could be a detergent. Now we know the importance of detergent. Since we know the COVID-19, we know the importance of ethyl alcohol. We know the importance of the washing hands by detergent, and we know that detergent even can kill COVID-19, right? So I don't have to give you any other example. So if it can kill a virus, it can kill bacteria and fungi also, right? So the disinfectants and in some cases antibiotics, I rarely recommend or use antibiotics because antibiotics create stress in plants, and many plants are susceptible for antibiotics, and antibiotics does not allow them to grow. Okay, so my suggestions, antibiotics only for animal cell culture, guys. But when you have something very critical, you can use low concentration of antibiotics for plant. When you have no other option to save your plant plate or plant material, then you can use antibiotic. Okay, a pre-wash, a small treatment, that don't grow them in antibiotic. Okay, because they become stressed, most of the plant. Okay. Physical treatments, autoclave, radiation, filtration, and ultrasonic babes. Where you see ultrasonic babes, it could be a burner, basically, where your spectacle, your scissor, your cutter, immediately you are going to sterilize. You don't have to heat them on the burner all the time, because when you burn it, you have to cold it. You have to wait like this. You are not doing anything. You just burn it, and then when it becomes red, you have to wait, because if you cut by this hot your eggplant, you will kill eggplant. So that is why ultrasonic waves become very beneficial. You just within 1 second you can sterilize the surface and can keep cutting in the laminar food, right? So disinfectant must have some compound that help them in the penetration throughout the cell membrane, cell wall of the bacteria and fungi. So detergent, for example, is the first and most important disinfectant for any kind of explant. So detergent has to be used. Okay. Now I used detergent from the Walmart, right? From the supermercado. They work very well. They truly work very well, but their efficiency sometimes is not very good. So we have to use molecular grade detergent like Triton 10, Triton 100, Triton 20. And these are the molecular grade detergent, which does not have any kind of contamination, because the detergent you are buying itself may have some contamination, right? So that is the one thing. What the disinfectants do, depending on the type. Very famous detergent and very favorite disinfectant that I like, it's chloro. Okay, you know chloro, Chlorex, chlorophenol, Chlorex, whatever you want to consider, they all works very well. Okay, and these are the one that enters in the cell wall and denature the proteins. Okay, or in some cases they decrease the activity of bacterial enzymes. What is the case of the antibiotics? They can also inhibit the growth and metabolism. They can damage the cell membrane, or they can even change the permeability of the membrane. So other things can also enter. So there are different kinds of compounds, and there are different properties. What detergent does? Detergent basically degrade the proteins and degrade the cell membrane, because cell membrane is made up of phospholipids, and phospholipids are soluble in the detergent, right? What alcohol does, I will let you know later. Okay. So for example, the laundry bleach is very good. My favorite, because it is not toxic. It can be used by 5 to 6% in volume, for like NaOCl. You can rinse thoroughly after the treatment, right? And like 2 times, 3 times by distilled water to clean it totally. And before it, you can also wash it with a detergent, right? So wash with the detergent, wash it with the distilled water, then wash it with the liquid laundry bleach like chloro, and wash it again, right? And then even you can use ethanol. Okay, so you have like 3 different treatment. So basically you make sure that your explant is totally sterilized. Okay, so I like this chloro bleach. I like this COCl2. Okay, this is also very good. It's a powder. And then we have ethanol, very, very important. 95% used to disinfecting plant tissues. Okay, so you can use from 80 to 95. Okay, in this case, okay, 80 to 95. This kills by bacteria by many means. It also degrade proteins. It also degrade the cell membrane. It also create the dehydration in the bacteria. Okay, so after like 10 to 15, 10 seconds to 5 minutes, the treatment can be given. 5 minutes, it could be 5 minutes, and then you can also again clean it by distilled water. Okay, so when you have these three treatment, detergent, bleach, alcohol, and you have rinsing the water throughout the process, then your explant is ready. Okay, so that is a normal process. Antibiotic can be used. I already mentioned there are long variety of antibiotic like carbonicillin, rifampicin, rifampicin, tetracycline, streptomycin. There are so many different kind of antibiotic that can be used. They are selective. Every antibiotic is specific. So it will never guarantee you that antibiotic is going to kill your bacteria, because now a lot of bacteria, they are already multi-drug resistant. Okay, normal bacteria even. So they have multi-drug resistance. So antibiotic will never provide you any guarantee until unless you use antibiotic which has multi-drug the capability, no? So the broad spectrum antibiotics, right? So my suggestion is not to use them. So that is why if you are using, obviously use them the broad spectrum. They did not induce resistance. They selectively toxic, and they preserve normal microflora. Okay, so depending on the antibiotic, I never use them. These are the process of each of these antibiotics that are used in the tissue culture lab. I'm not going to give you any details. You have to read it. Okay, so they are inhibitors of the protein synthesis, inhibitor of the cell membrane, and inhibitor of cell wall synthesis. Okay, so these are like three categories. And there are some categories that are inhibit the metabolites, and some of them they inhibit the nucleic acid. Okay, so in total you have 5 categories. There is another chemical which is mercuric chloride. I don't like it much because it's very toxic, and sometimes people who are not very good in working in the lab, they can inhale, they can mishandle the compound, and it can be damaging. So, but yes, it's very, very effective. It's very toxic. It's very effective. So it denatures the proteins, but in many lab they use it because it's guaranteed the surface sterilization. It guarantees the surface sterilization. So that is why people use it. I don't like it because I avoid these such amount of toxicity in the lab. Okay. Okay. Another factor is the UV radiation. You know that radiation, UV light, create mutations. Okay. So when we are always in exposure of UV light, always. If you are walking, suppose if you are walking from Tec de Monterrey to Centro in a day in a sunlight, or you walk on a beach, any beach in a full sunlight, 5 kilometers, you will have more than 100 mutation in your genome. Okay, naturally. And these mutations are caused by the UV light. Okay, thanks to our non-coding reason in our body that protect us to have these mutations have a negative impact on our health. Okay, but when you have this UV light in a full intensity and a spectrum by this range 200 to 400 nanometers, and you have a very close surface on it, definitely the intensity is going to be very aggressive, right? So for example, the best wavelength to get the UV light is 254 nanometer. And what it would create, this is the UV range. So basically, you remember the blue and red light, right? For the photosynthesis, and the UV light have the killing efficiency. And how does it kill the bacteria and microorganisms? By mutations. It creates massive amount of mutation on the genetic material, whether it is DNA, whether it is RNA. And when the genetic material is mutated with a significant fractures and damage, it will definitely distort the whole central dogma. So they will not be able to write proteins, and eventually the microorganism, bacteria and virus will die. Okay, so this is how UV work. Now, another method is autoclave, right? Many people have already knowing about autoclaves, all that, right? That looks very simple. But how the autoclave works, okay? So before the autoclaves, this technique comes in the category of heating using the temperature, right? For example, few things like oven. Some ovens can be used without using autoclave because autoclaves, it's a time consuming process. So basically you have an oven that can reach up to 100 degrees centigrade. You can put 3 hours, your stainless steel material at least, right? They will not have anything with this temperature and everything will be sterilized. Microwaves, the normal microwaves, domestic micro oven or microwave can be also used to sterilize some of the equipments. Okay, it does not guarantee. Most of the time you have to sterilize how long, how much period of time you have to microwave. You cannot put metals, right? You can put dish, you can put flask, you can put media. I never use microwave. I use microwave for other purposes, for extraction and all that, but I never sterilize anything in microwave. Flaming and heating of the tools, direct flaming, right? In laminar flow, you know, you always have a fire lamp there. So you are using the dry fire all the time to clean the surface of the stainless steel scissor and scalpel and other things, right? Another method of heating is I already mentioned is the autoclave. Autoclave, the main principle of autoclave is very interesting, and generally people do not know how the autoclave works. Anybody who can explain me autoclave, how it works from this group? I'm listening. Yes, Emilia.
Well, with the autoclave, it works with steam, with really hot water. You have to put batches for 15 minutes and it reaches a temperature of 121 Celsius degrees. And that's how you will sterilize many different kind of things. For example, a material like, I don't know, for example, the media that you're preparing, you have to sterilize with autoclave. So I think that's
okay. So thank you, Amelia. Good. The most important thing in this whole technique of autoclave is steam is the main key player, because steam, you know, has more heat than the heat. You understand what I'm point is? If you put your finger in a candle, you can put it like this. This is the candle, right? This is the lighter. Okay, this is the lighter. You see my lighter? You see my fire or not? Oh, I cannot see fire. Why? You see fire? Oh yeah. So I can put my finger like this. Do you see this? You see or not? I'm putting my finger, nothing is happening. But if I have the steam here, I steam spray, do you think I can put like this? No, because the penetration power of the vapor particle is extremely energetic. So that is why the pressure combined with the water and temperature create pressure with the steam that is deadly. So within seconds, it is going to enter in any living cell inside that environment with the temperature and pressure and the steam, and it will get everything. Okay, so that is the main principle of the autoclave. Autoclave could be 15 minutes, it could be 20 minutes, it could be 25 minutes, depending on the volume, even 40 minutes if you are autoclaving something which is 1000 or 2000 liter, right? So depending on that, it's going to change, but this is a standardized. This is the standard 121 degrees centigrade, 50 lbs per square inch the pressure. Okay. This is I already mentioned filtration. So the filtration, what you filter and how you filter. We have paper filters normally in a tissue culture lab. Whatman is a very famous company. They produce world's best filter paper, Wattsman. Okay, and Wattsman is a very company who has dozens of patents in that area. So they create different kinds of papers, basically using suction, using a flask. You can put them between the flask and you can autoclave whatever. You can sterilize anything you want, which is liquid, which is not in temperature, which are heat sensitive. So solution like vitamins, solution like hormones, phytoregulators. They are the main compounds that are used for the sterilized by the filtration. Okay, and that is very important. Don't you cannot autoclave them or you can heat them. They are even more than 40 degrees centigrade. They will basically degrade. Okay, they will lose their functionality. Okay, so that is the filtration of the liquids. Now filtration of the hood, laminar airflow. Everybody know that, right? Laminar airflow. What is the principle of laminar airflow? Anybody who can tell me? I'm listening. Campana de flujo laminar. Who can tell me the principle? How does it work? Yes, Amelia.
Well, it's actually literally like a box that is filtrating with a filter called HEPA, I think. And it's a constant airflow that it's sterilizing or filtrating the contaminants that it may have when you put your equipment inside of there.
Okay, thank you, Amelia. The main objective of laminar airflow is to create sterilized air, right? It creates air which does not have bacteria. So at the same time, when it is creating the air, this air has some force. So the surface where you are working, it is pushed by the sterilized air. So means that anything outside is not entering. So it has a velocity like 20 to 30 kilometers per hour of air. So basically, whatever is moving outside, it's not able to enter. So what you are creating a complete aseptic, sterilized environment. And obviously, it has filters and other things that I will explain. Okay. This is just a small picture of first autoclave. Okay, this is in the Museum of Paris. Okay, Paris Museum, you can find this first autoclave. It does not have, you know, electricity. This is just because of the fire, and this works very, very manual. It was developed by Charles Chamberlain, a French scientist. Okay, it was developed in 1879. So you can imagine how people used to autoclave or sterilize things before that year, because tissue culture is so old. In 18th century, there were so many attempts were made, but you can imagine how people were trying to create aseptic condition. Right now, you have different kinds of model. This is our this model we have in our laboratory. So basically, it's like 10 liter of capacity. It's completely automatic. It will give you signals. It will give you the pressure and all the indicators. Okay. And we also have very big size of autoclaves. It could be 1,000 liter, 2,000 liter. They are very expensive, right? So this is one thing. And we were talking about the laminar flow. Laminar flow has a filter. This is called HEPA. Okay. Now HEPA is very common. I buy this section. You see this is the I forget the aspirador. Okay. This is the small aspirador. It also has the HEPA filter inside. Okay, so HEPA is now very common. It is being used in the air conditioner. It is being used in the domestic things also. But the HEPA filter, what you use in a laminar flow, it has many layers. It is the best HEPA filter that you can think about, that can filter and trap 99.97% of particles of bigger than 0.03 micron. Okay, so anything lesser than 0.3 micron can pass out. Bigger than 3.3 microns, it's going to be outside. So basically, you are going to create an environment that is free from microbes. And HEPA is called high efficiency particulate air. Okay, and these young ladies here, it's just to represent, for example, the vertical and horizontal the hoods. For example, here you are seeing horizontal. Okay, here when you have air front, okay, on your directly on your face. Okay, and in this case, this is a vertical where the air is being produced from the top. It falls on the floor and then it's turned on your face. Okay, when you're working. I personally have some psychological problem with this. And I will share this because I never liked this one. I don't know why. Okay, I always prefer the direct vertical, sorry, the horizontal laminar flow when you have constant flow from directly in front of your face. Okay. In this case, when it is coming from top and then turning around, basically I sometimes think that maybe there is not enough pressure or something like that. So I always buy or prefer these horizontal laminar flow. Okay. So laminar flow, there are different types. One very small size, very big size. The biggest size is then 3 people can work together. Okay, but it is not allowed more than 2 people. Okay, so the size is depending on that. There is, you have HEPA filter, the ventilator, and you have the pressure. That's it. And this is the glass shield. This is the glass shield. Always try to buy or prefer the covered. Okay, because you protect them. Once you complete your work, you will close them and you maintain them clean. Okay, so here in this case, for example, there is a UV light lamp. Okay. Upside here, UV light lamp. When you open UV light before working like 30 minutes, you can switch on and don't work. Just switch on before working. When everything is got sterilized, you go put the alcohol on the surface and then start working. Okay, you have to switch on since the UV is open. Okay, and when it is switch on, means you don't have to close the lid. Lid should be open. Lid should be open and it should allow the ventilation. But if you only want to put the UV on, then you can close the lid. Okay. So most of the time, what people prefer, they put 30 minutes the UV on, and then they switch on the hood. Okay. They turn on the ventilator and switch off the light. Okay. And still they wait few minutes. Then after they start working. Meanwhile, they clean all the surface by 70% alcohol. Okay, so these are the good practices. How alcohol kills the bacteria. Alcohol basically degrade the membrane, okay, of the bacteria, and basically there are 2 kinds of bacteria: gram-negative and gram-positive. Gram-positive bacteria have a very thick layer of a carbohydrate molecule that is called peptidoglycan. Okay, so depending whether it is positive or negative bacteria, they will be killed by little different timing. Okay, the gram-negative bacteria, which has a very thin layer of peptidoglycan, allow alcohol pass quickly. So when it enters, it denaturalizes the proteins and kill the bacteria. When it is thick, it will take few minutes more. Okay, few seconds and few minutes more, depending on the bacteria. Now, my question for you: why you use 70% alcohol and not 10% or 50% or 100%? I'm listening. Emilia, you already answered so many questions. Let me wait some other people to reply, then I will come back to you. Okay. Yes, Stefania.
I don't know if it's the truth, but I think it's because if it's 100%, it will evaporate.
No. Any other answer? Are we sleeping? Are we listening? Are you there? You never think about, since we are in COVID, like in pandemic, since year and a half, and you never thought why you are using 70% alcohol? Come on. Yes, Carla.
Because it can help destroying the bacteria cells on the contaminants, but it doesn't damage the ones you want to study.
Now, Pilar.
I was thinking the same thing as Carla.
Okay.